There are multiple different types of stem cells. A general definition which can include all of the different types would be as follows. Stem cells are cells that are not fully differentiated. When we say differentiated, we mean specialized. Our bodies are made up of many different types of specialized cells. We have lung cells, liver cells, heart cells, and many other cell types. All of these different cell types contain the same DNA. What is different between the types is which genes are turned on. Stem cells are cells whose fate has not yet been decided. Some of their genes are turned on, but they haven't turned on the specific genes that are needed in order for them to become some specialized type of cell. Much of the controversy associated with stem cells has to do with where they come from. In this video, we will review sources of stem cells. The three major types of stem cells which have been and are currently being investigated by the scientific community are embryonic stem cells, adult stem cells, and cord blood-derived stem cells. We will discuss embryonic stem cells first. Embryonic stem cells are pluripotent. This means that they are capable of becoming any type of cell except for cells which support the growth of the fetus, for example, placenta. There are three ways in which we can obtain embryonic stem cells: through in vitro fertilization, through therapeutic cloning, or from already available stem cell lines. We will discuss in vitro fertilization-derived embryonic stem cells first. In vitro fertilization, an egg and sperm are allowed to fuse outside of the body. The term fertilization means fusion of the sperm and egg, and in vitro means in glass. The fertilized egg is then implanted into the woman's uterus and grows into a baby. This is done to help couples who have trouble conceiving. Oftentimes, there will be several embryos left over that the couple no longer needs once they have successfully become pregnant, and these embryos will be thrown away. Instead of being thrown away, they can be used to harvest embryonic stem cells. Once fertilization occurs, the new embryo begins to divide. It divides until it becomes a hollow ball of about 100 cells called the blastocyst. The blastocyst contains an inner cell mass, and these cells are the embryonic stem cells, which can be individually dissected out of the blastocyst and then grown in a petri dish. The blastocyst as a whole is totipotent. This means that it can make any cell type in the body and also make cell types required to support the growth of a fetus, like placenta. The embryonic stem cells which came from the blastocyst are pluripotent. As we stated earlier, we can also obtain embryonic stem cells through therapeutic cloning. Therapeutic cloning is also known as somatic cell nuclear transfer. In therapeutic cloning, the nucleus of an egg is removed and replaced with the nucleus of a somatic cell. All body cells are called somatic cells, so this could be a skin cell, lung cell, heart cell, or any other type of body cell. Egg and sperm are called sex cells, and they are not the same as the rest of the body cells. The new cell is then exposed to an electrical pulse which induces it to begin dividing. The new cells divide until the blastocyst is formed, and the inner cell mass can be harvested the same way as can be done with in vitro fertilization embryos. Embryonic stem cells made this way are genetically identical to the donor somatic cell. As we mentioned previously, there is one other way in which we can obtain embryonic stem cells, and that is to obtain them from other researchers or research organizations. There are several stem cell lines which have already been created that can be accessed. So, each researcher does not have to make their own stem cell line in order to do stem cell research. The next major stem cell type is the adult stem cell. Adult stem cells can be removed from the blood, bone marrow, retinas, or some other organs. Adult stem cells are multipotent. This means that they can make certain cell types, but not all cell types. For example, blood stem cells can make any type of blood cell, but they can't make skin, heart, liver, or lung cells. They are not as versatile as embryonic stem cells, and it is generally necessary to use invasive methods to obtain them. However, because harvesting adult stem cells does not require the use of embryos, their use is much less controversial than the use of embryonic stem cells. The third major type of stem cells is the cord blood-derived stem cell. These are stem cells that are harvested from umbilical cord blood after the baby is born. Like adult stem cells, cord blood-derived stem cells are also multipotent. Specifically, cord blood-derived stem cells can make any type of blood cell, but they can't make any other kind of cell. The three stem cell sources which we just described are the sources that have been and are being used today. There are three other types of stem cell sources which have only very recently begun to be explored, and we will just briefly touch on them. The first of our three additional stem cell sources is the dead or growth-arrested embryo. These are embryos that were produced by in vitro fertilization technology. The egg and sperm were allowed to fuse and the embryos began to divide, but for some reason, some of the embryos simply stopped growing. Researchers waited for 24 hours to see if the cells would start dividing again, and when this did not happen, they declared the embryos dead and dissected out some of their cells. The idea here is the same as when we use organs for transplants from people who have died. Some of their organs are still functional and can be used by other people. These embryos died, but some of their cells were still functional. The problem with this stem cell source is that these embryos may have stopped developing because there was something wrong with them genetically, and so it could be dangerous to try to use them for any kind of therapy. The aborted fetus is probably the most controversial stem cell source. Research has shown that blood stem cells can be found in aborted mouse fetuses. These are stem cells that can make any type of blood cell, but no other cell types. Further studies in animals will be required to determine whether or not there are any more useful cells in these aborted fetuses, but the current research suggests that these stem cells are not very different or more useful than adult stem cells or cord blood-derived stem cells, which are less controversial. It is very unlikely that we will use this as a major source of human stem cells in the future. Two groups of scientists, one in Wisconsin and the other in Japan, have recently published research showing that skin cells can be reprogrammed into pluripotent stem cells. The research was based on what is known about therapeutic cloning by somatic cell nuclear transfer. During therapeutic cloning, when the nucleus of an egg is replaced with the nucleus of a somatic cell, certain protein factors within the egg reprogram the somatic cell nucleus so that the new cell is undifferentiated. These scientists added those protein factors to skin cells, which caused those cells to de-differentiate, or become undifferentiated, into pluripotent stem cells. This work is still very new, but it is looking very promising as an answer to the stem cell debate. This is currently the only method for generating versatile, pluripotent stem cells without using embryos.